All right, welcome back to the deep dive. Today we're heading into the complex world of urban ecology.
That's right, and we're doing it by focusing on just one key scientific paper. It's a great shortcut to get up to speed on how cities are changing aquatic life.
Exactly. We're looking at a paper by Zhang and colleagues. It was published in Science Advances.
And the title is, let me get it right, Environmental DNA Captures Native and Non-Native Fish Community Variations Across the Lentic and Lotic Systems of a Megacity.
Quite a mouthful, but it tells you a lot. Okay, so let's unpack it. What's the core problem here?
Well, it's pretty huge. We know urbanization is a major driver of biodiversity loss globally.
Right, cities expanding everywhere.
But figuring out what's happening specifically to fish, the main vertebrates in these water systems, is really, really hard, especially in dense urban areas.
Yeah, I mean, how do you even survey fish properly in, say, a small city park pond or a heavily managed canal? Traditional methods like netting just don't work well there.
And it really matters. Fish are critical indicators, you know. Their health tells us about the water quality overall. Plus, they play a big role in how nutrients move through the system.
So understanding how cities impact them is crucial for planning more sustainable cities, especially megacities like Beijing, which was the focus here.
Beijing's a perfect case study. Massive city, over 21 million people, rapidly changing waterways.
And the researchers pointed out a key gap, didn't they? Most studies only look at rivers and streams, what scientists call lotic systems.
Exactly. They often ignore the still water bodies, lakes, ponds, reservoirs, the lentic systems, which are actually pretty common in cities.
If you're only looking at the rivers, you're missing a big piece of the puzzle.
A huge piece. You're potentially undercounting the total fish diversity quite significantly.
Okay, so that brings us to the method. How did they get around the challenge of surveying fish across, what was it, over 100 different sites in Beijing?
Yeah, 109 sites, a mix of lentic and lotic. And they used a really neat technique, environmental DNA metabarcoding, eDNA.
Ah, eDNA. So this is the DNA that organisms just shed into their environment.
Precisely. Fish are constantly releasing DNA through skin cells, waste, mucus, you name it. It ends up floating in the water.
So you just take a water sample.
Basically, yeah. Take a water sample, filter out the DNA, and then sequence it. Using genetic databases, you can identify which species that DNA belongs to.
That sounds like a game changer for city surveys.
Oh, absolutely. It's non-invasive. You don't have to catch or disturb the fish. It's incredibly sensitive, picking up even rare species. And it lets you cover a massive area cost-effectively.
Which is how they managed 109 sites. Right?
Yeah.
Covering the whole gradient from the city center, they used Tiananmen Square as the reference point.
Yeah, distance from Tiananmen Square was their index for urban intensity. They sampled all the way out to the more rural edges of Beijing.
A really comprehensive snapshot then.
The first time something like this has really been feasible at this scale in such a complex urban environment.
Okay, so what did this eDNA suite actually find? What was swimming in Beijing's waters?
Well, they recovered genetic signatures for 75 different freshwater fish types. Technically, molecular operational taxonomic units, or MOTUs.
75, wow. And how did that break down between native and non-native species?
It was about 52 native types and 23 non-native.
So more natives than non-natives overall. That sounds...
On the surface, maybe. But here's the kicker. Those 23 non-native taxa, they were found in 92.5% of all the sites they sampled.
Wait, 92.5%. So invasive or introduced species are basically everywhere.
Pretty much everywhere. We're talking things like channel catfish, North African catfish, largemouth bass, tilapia species often introduced for aquaculture or fishing. They are pervasive.
That's sobering. Okay, what about the difference between the ponds and the rivers? The lentic versus lotic.
That was interesting. The lentic systems, the ponds and lakes, actually supported significantly more native species per site compared to the rivers and streams.
So those still water bodies are acting as important refuges for native fish.
It seems so, yeah. They hold higher native richness, but there's a flip side. When they looked at how different the fish communities were between sites, what we call beta diversity, the lentic communities were much more similar to each other.
Ah, so more homogenized. Less unique from pond to pond.
Exactly. The lotic systems, the rivers, showed greater variability between sites. This suggests the pond communities might be more ecologically stressed or isolated, losing their distinctiveness.
So ignoring ponds really does skew the picture. They hold more native species, but might be more vulnerable in other ways.
Definitely. You miss a lot of the native diversity count if you skip the ponds, but you also miss this pattern of homogenization.
Okay, this is where it gets really interesting, I think. They tried to figure out what environmental factors were actually driving these patterns in fish diversity.
Right. The usual suspects in urban ecology are things related to the physical landscape.
Like how much concrete is around, the percentage of impervious surfaces, right? Or lack of vegetation.
Exactly. Impervious surface and vegetation cover, percent veg. Those are standard measures of urbanization intensity.
But that's not what they found driving the fish patterns.
Surprisingly, no. Those landscape variables showed very little effect on fish diversity in this study.
Wow, okay, so if it wasn't the concrete jungle aspect, what was the main driver?
It was water quality. Specifically, one measure stood out, chemical oxygen demand, or CO.
CO, right. That's a measure of organic pollution, isn't it? Like sewage or industrial waste.
Precisely. It indicates how much organic gunk is in the water, using up oxygen as it decomposes, and that was the single most influential factor they identified.
So pollution over pavement.
Seems like it for the fish communities here. High SWAD had a strong negative impact on the richness of native fish species.
Fewer native species where the water was more polluted. Makes sense.
Yeah, but crucially, this negative effect was much, much stronger in the lentic systems, the ponds and lakes.
Because they're isolated, the pollution just sits there.
That's the thinking. Rivers can flush pollutants downstream to some extent, but ponds act like sinks. The pollution accumulates, oxygen levels drop, and the sensitive native species just can't survive.
But the non-natives.
Often, they can tolerate those stressful conditions much better. Many introduced species are chosen for aquaculture because they're hardy.
So they have a built-in advantage in polluted city waters.
Exactly. They can outcompete or replace the native species that are struggling. The study gives an example. A sensitive native minnow, Zachoplatypus, was almost gone from high sod sites.
While the invasive catfish or tilapia were probably doing just fine.
Their occurrence didn't seem to change much based on the CO level. They were equally common in cleaner or dirtier water. It really highlights how pollution filters out the natives and favors the invaders in these static systems.
Now, there was another finding that really seemed counterintuitive, especially concerning the rivers and streams.
Yes, the lotic gradient finding. This one definitely goes against some common assumptions.
Usually, you'd think invasive species would be most dominant in the highly disturbed city center. Right.
That's the typical pattern often reported. More disturbance, more human activity, more introductions leads to higher non-native abundance.
But in Beijing's rivers, they found the opposite.
For the relative abundance of non-natives, yes. It actually increased as they moved away from the city center towards the more rural areas.
So, more non-native fish further out in the less urbanized parts of the rivers. How does that work?
Well, the researchers suggest a couple of things. One idea is about habitat. While city center rivers might be heavily channelized and polluted.
Like concrete canals.
Right. The rivers further out, even if still impacted, might offer more diverse habitats, more natural banks, varied flow, places for fish to hide and thrive, more structural heterogeneity.
Okay, so maybe better real estate for non-natives to establish themselves once they get there.
Potentially, yes. And the getting there part connects back to connectivity. Rivers are, well, connected.
They flow.
Exactly. So once non-native species get into a river system, they can spread relatively easily along that network, colonizing new areas, including those more structurally diverse rural stretches.
Unlike the ponds, which are like isolated islands.
Precisely. The ponds limit dispersal for everyone, natives included, potentially leading to genetic isolation. But the rivers can act like highways for non-native dispersal.
So it's a really different dynamic depending on the water body type.
Completely different. And it's important to remember, even with all these factors, their models only explain about 55% of the variation in species richness.
Meaning there's still a lot we don't know, other factors involved.
For sure. Things like water flow speed, the amount and type of aquatic plants, specific human disturbances, lots more to investigate.
Okay, so pulling this all together, what are the big implications here? What does this mean for managing city waterways?
Well, the clearest message, I think, is that you can't have a one-size-fits-all conservation strategy for urban waters.
Because the challenges are different in ponds versus rivers.
Exactly. For the lentic systems, the ponds, the lakes, the absolute priority has to be managing pollution. Tackling that high sod, controlling wastewater inputs. That's what seems to be hammering the native species trapped in those systems.
Okay, so Job 1 for ponds. Clean up the water. What about the rivers?
For the lotic systems, while pollution still matters, The bigger, perhaps more unique challenge highlighted here is managing the spread of non-native species.
Especially because they seem to thrive further out, where the river habitat might be better.
Right. So you need a really stringent monitoring and potentially control efforts focused on those non-native populations, particularly in the connected river networks where they can spread rapidly.
It's a much more nuanced picture than just cities are bad for fish.
Definitely.
And we should probably touch on the limitations of eDNA again briefly. It's powerful, but not perfect.
True. While it tells you what species are present, getting accurate abundance, like how many fish are there from the amount of eDNA, is still an active area of research. There isn't always a simple correlation.
And the transport issue, DNA flowing downstream in rivers.
Yeah, that's consideration. DNA detected at one point could potentially have come from upstream. The study tried to mitigate this by spacing their river sites far apart, more than 3 kilometers, but it's something to keep in mind when interpreting results from flowing water.
But the potential for the future seems huge, especially with citizen science.
Oh, absolutely. Because taking a water sample is relatively easy, non-invasive. You could potentially equip community groups, schools, local citizens to collect samples across a whole city.
Imagine the amount of data you could generate for monitoring.
It could revolutionize how we track the health of these ecosystems and see if our management efforts are actually working in almost real time.
So, wrapping up, what's the main takeaway from this deep dive into Zhang and colleagues' work?
For me, it's the crucial reminder that urbanization impacts life differently depending on the habitat. What happens in a city pond is driven by different pressures than what happens in a city river, even within the same urban landscape. Pollution versus connectivity, for instance.
We can't just lump urban waters together.
Not at all. The type of water body fundamentally shapes the ecological challenges.
Okay, that leads us to our final provocative thought for you, the listener.
Yeah, thinking about this, we know cities often create new lentic systems, right? Things like stormwater ponds, ornamental lakes, reservoirs, they're often isolated, man-made.
Right.
So the question is, instead of just building these isolated ponds and focusing only on water quality after the fact, could city planners actually design new urban water bodies proactively to enhance connectivity for native species?
Building in connections from the start, like fish ladders between ponds or designing them as part of a linked network.
Maybe. Could we engineer urban water systems not just to hold water, but to actively support native biodiversity movement and resilience in the long run? It shifts the focus from just mitigating harm to potentially designing for ecological benefit.
A really important question as our cities continue to grow, something to definitely think about. We hope this exploration of urban fish and eDNA has given you some new perspectives. Thanks for joining us on the deep dive.
Until next time, keep exploring.